So, hello everyone. We are once more in the What We Are Reading section or video blog of the South American Mycorrhizal Research Network. This time around, we have Laura van Galen from ETH Zurich in the Thomas Crowther Lab. She's a postdoc there. We also have Professor Nati Fernández from Universidad Nacional del Comahue, right?
Right.
And me and Laura authored this really interesting paper, which I will show in a few seconds, in Phytologist, entitled Correlated Evolution in an Ectomycorrhizal Host-Symbiont System. This work was done in New Zealand, and the reason that Nati and me are present today is because the paper was done in a Nothofagus system, in 5 Nothofagus species. So both Nati and me are very, very interested in this kind of system. So my first really basic question to you, Laura, would be, what was the main question behind this article, and how this research came about?
Yes, so this research was part of a bigger project that I did for my PhD at the University of Otago, which was about understanding the ecology of ectomycorrhizal fungi in Nothofagus forests. And so the main aims of this paper was to look at how specialized different ectomycorrhizal fungal communities are to different host species, because there's been lots of research showing that whilst there are many generalist ectomycorrhizal species, often they do show preferences to different host families or different host genera, but there's less evidence of whether those preferences also occur to more closely related hosts, like within the same genus. And then the second aim was to investigate how the evolution of the hosts and the symbionts might be related, because we know that ectomycorrhizal fungal species are hugely diverse. They're a lot more diverse than other groups like the arbuscular mycorrhizal fungi. And so we wanted to look to see whether this specialization might be leading to diversification within fungal lineages. So looking to see whether or not the phylogenies of the hosts and their symbionts might be correlated.
Okay, so which were like the main results of your study?
Yeah, so to do this, we used the Nothofagus in New Zealand, which there's 5 different Nothofagus species. And well, perhaps I should first explain that the Nothofagus also belong to different subgenera, so they can be divided up. And there's 4 different subgenera. And so in New Zealand, we have 1 species from the Lophozonia subgenus, which is the oldest, and then we have 4 species from the Fuscospora, which is one of the newer ones. And so we've got this sort of differences in the relationships between the hosts. And so I was sampled fungal communities within each of these 5 different host species, and then built a fungal phylogeny and looked to see how related those 2 groups were. And we found, really interestingly, that there was a lot of specialization going on. So nearly a quarter of the fungal species looked at showed statistically significant preference towards a particular host species or one of the subgenera. And when we looked for tests of the correlated phylogenies, they were significant to quite some substantial phylogenetic depth. So it's really exciting to see that there's this relationship that's phylogenetic and evolutionarily based between these 2 groups. Because we often see this in host parasite systems that are quite closely linked, but it's super exciting to see that it's also occurring in these mutualistic relationships that are slightly less specific.
Excellent. I think that in part you answered my next question, which would be why Nothofagus and how many species do you have? I think that you already mentioned that, but
well, yeah. I mean, I think Nothofagus is a really interesting group to be looking at because it is so widespread across the Southern Hemisphere. So it's in New Zealand, but also, as you guys know, in Southern South America, but in Southeastern Australia, New Caledonia, New Guinea, and so it's back from when the land masses were joined for Gondwana. So it's something that's really, it's a group that's really of interest to our research group at Otago. And it's very widespread across New Zealand as well. And so people for years have been researching the fungi in these forests, but mostly through collecting fruiting bodies, doing surveys. And so we really, up until now, haven't had a good understanding though of what's in the soil below ground, which is where the vast majority of fungal species are. So really this project came about because we wanted to learn more about the fungal species in these forests in New Zealand.
I completely agree with you. I love Nothofagus forests.
Yeah,
and I wish
they're very beautiful
sometime
to work in.
I can visit your forest, your Nothofagus forest. So,
Yes.
do you have any other native ectomycorrhizal forestry species in New Zealand, or only Nothofagus?
No, we have some other ones. So there's the Leptospermum genus and the Kunzia genus. They're also ectomycorrhizal. And there's a couple of species of Pomaderus, but that's it in terms of native hosts. And Nothofagus are by far the most widespread. But of course, these days we have a lot of Pinaceae species and eucalyptus species that have been introduced to New Zealand, and they have also brought ectomycorrhizal fungi. So for my project, I specifically avoided those areas and just sampled in pure Nothofagus forests. But yeah, the dynamics of ectomycorrhizal fungi are changing a lot because of these introduced plants.
Great.
So that brings me to my next question regarding exactly the sampling scheme. You had, I just want to show the map because it was an amazing.
Yes, so many different sites.
So many.
I must say
different
it was excellent field work to conduct.
Yes.
Here you can see the map of New Zealand and the 5 different species of Nothofagus and different sampling sites, 81 sites. So I wanted to ask you, why did you use in-grow bags instead of root tips or instead of rhizospheric soil? Why
Yeah.
this specific method?
Yeah, so I sampled the fungi using the high-fill ingrowth bags, which are small little bags made of mesh filled with sand. And so the mesh is very fine. And so fungal hyphae can grow inside, but roots can't. And so the idea is you bury them in the soil, leave them there for months. The fungi will grow inside the bags, and then you can come back and take them up again. And then you've got a little packet of fungal hyphae to extract the DNA from. And so there are lots of methods to sequence fungal DNA, and one is collecting soil samples or root tips, like you said. But we chose ingrowth bags because for soil samples, you're sequencing everything in the soil, so including spores, dead fungal material, everything that's there. So it's not necessarily the species that are connected to the hosts at that moment. And you also need a huge sequencing depth to be able to detect the ectomycorrhizae that we're interested in. But root tips are also great because you're actually sampling the mycorrhiza themselves, so you know that the connection is there. But we chose ingrowth bags because they're a really efficient and easy thing to use. All you have to do is bury them, leave them, and come back and collect them. So in terms of sampling, it meant that we could sample over a much wider area. And also, there have been people who've researched the difference between different methods, and some of them have found that often you can miss things on root tips because they tend to be dominated by some really common fungal species, and it's harder to detect the rare things. Obviously, we're not detecting everything with the ingrowth bags also because they're biased in a different way. Some fungal species might prefer not to grow into them. So no method is perfect, but I think that ingrowth bags are great because they're sampling the things that are actively growing, so things that we know are alive and operating within the ecosystem, and they're also just so efficient to use.
Great, thanks. Nati?
Perfect. Yes. So could you briefly explain how you came out to this sampling design? I mean, how did you select the places? I mean, did you sample in all the Nothofagus forests you found, or you selected them somehow? And why did you decide to select so many different places instead of taking more samples from each forest?
Yeah, so I wanted to sample as a wider range of these forests as possible. So Nothofagus, they span their structure is very different. They can occur in some places right from sea level through up to the tree line, or across huge gradients of rainfall and moisture, very wet, very dry areas. So I tried to stratify sampling across altitudes and climate and space. And so we were pretty much limited by what was accessible by car. So I spatially distributed a heap of target sites and then went and visited them. And if they matched the criteria, if they were Nothofagus forests that didn't have other ectomycorrhizal species nearby, then we sampled them. And so we took a lot of samples within each of these 81 sites. So within each site, we set up 10 plots, and within each plot, we buried 5 hyphal ingrowth bags. So there was 50 bags at each site, because there is huge spatial variation in fungal communities in the soil. So we can't just sample from one spot because it won't be representative of that area. But then I ended up pooling the ingrowth bags and then the data from the different plots to kind of characterize the community at that site. And so, most of the analyses were done at the site level. So having 81 sites was good to be able to investigate patterns occurring across the environmental and spatial gradients.
Laura, so this for me was one of the most interesting results related to phylogenetic diversity among the different Nothofagus species. I wonder, so you clearly show, for example, that the relative, what is the difference between relative phylogenetic diversity and like total phylogenetic diversity?
Yeah, so the phylogenetic diversity there is how phylogenetically specialized the communities are. So this is looking at the 4 main host species. And so the first one, Nothofagus menziesii, is from the Lophozonia subgenus, and then the other 3 are from the Fuscospora subgenus. So there's that divide there. But these are Z scores. So the points below the dotted line, if the confidence interval doesn't touch the dotted line, it means that it's significant. And so for all 4 of those species in A, we're seeing that they are significantly phylogenetically specialized. So they're more closely related than we would expect if we were to just randomly sample from the tree. The relative phylogenetic diversity is a measure of the branch lengths. It's kind of hard to explain, but points above the dotted line indicate that the branch lengths are longer than we would expect if we were to randomly sample. And points below the line are when branch lengths are shorter. And so these differences weren't statistically significant, but we see this really awesome trend where the Lophozonia host, the Nothofagus menziesii, has longer phylogenetic branch lengths than the other. So that's super interesting because we know that the Lophozonia is the older host, so perhaps that's indicative of a much older phylogenetic association between the hosts and their symbionts. There's been a lot more time for that diversity to accumulate. So it's possibly a bit more complicated because when we looked at just the richness of the ASVs, we didn't see such a strong pattern. There was a trend, but it wasn't significant. So it's complicated, but we need more investigation about that.
One last question about the results. Did you maybe examine the relationship between the distribution, like the geographic range of each of these species and the diversity of ASVs that you found?
So there wasn't the spatial proximity of sites. So I ran another model to try and compare how much variation in the fungal communities was explained by the host trees versus the spatial proximity between sites. And much more variation was explained by host. There wasn't a lot of difference between space.
I wonder. No, go ahead. Sorry.
Yeah. So in a separate paper to this, also with this data set, we were investigating more specifically the factors affecting the community turnover, so fungal community assembly, like deterministic factors versus the stochastic factors. So comparing the effects of environmental host versus spatial stochasticity, like dispersal ability. And we found that by far the communities were much more influenced by the environment and their hosts compared to the spatial proximity.
I wonder, and this leads to the last question that Nati will make. I ask this because here in South America, some species, some Nothofagus species, like maybe Nothofagus alessandri or Nothofagus glauca, have a very restricted distribution, while others like Nothofagus pumili and Dombeji have a huge distribution. So, Nati, go ahead with the last question.
Yes, and this is one of the most interesting questions, at least for me. And is, do you think that we will find like similar patterns or tendencies in our Nothofagus forest here in South America? We also have some species comprising the Lophosonia subgenus, for example.
Yeah, I think that would be the perfect test to see, to look at the South American Nothofagus species and see if we see the same pattern, because I think that there's every reason to expect that we would. And also, I think you also have hosts from the subgenus Nothofagus as well as Lophosonia and Fuscospora. So there'd be 3 subgenera to look across, which is a wider range. So I think that will be really interesting. And yeah, I mean, I can't imagine, I think the processes operating in South America are probably quite similar over evolutionary timescales than what's operating in New Zealand. So it'll be really exciting to see if it's a similar pattern.
That could be the next step.
Absolutely, yes, yes. And also, I guess, to look at other major ectomycorrhizal host genera like the Eucalyptus or Quercus or Pinus and see whether or not we're seeing similar patterns.
Yes, that's true. So it was very interesting.
Yes.
Thanks very much.
Excellent. Thank you so much, Nati. Thank you so much, Laura, for a very interesting discussion, and we will see you next time. Okay, bye.
Bye.
Bye.